The last thing that we looked at in the degradation of amino acids was the enzyme aminotransferase. Aminotransferase is a complex set of reactions that occur where you have two stages. One, you take the amino acid in, and the purpose of the aminotransferase is to transfer the amine. So the carbon chain and the amine are taken care of in two separate places. The aminotransferase is going to take the amine, and it will react it to the enzyme and attach it to the PLP cofactor. And at the end of the reactions, and I showed the mechanism all the way up to this point in time, so when we look at it, here is the alpha keto acid, there's the keto, and alpha to it is an acid, and the R group, which is dependent upon the amino acid you're looking for, or you're looking at, and the amine that was originally on there. Now, in the second stage, we can bring in any alpha keto acid, but normally it's alpha ketoglutarate in the incoming position, and the amine that was transferred from the amino acid we're degrading gets implanted or placed into the alpha keto acid of alpha ketoglutarate, and the amino that comes out most often is glutamate. Now, one thing that we do look at and know about aminotransferases, other than the mechanism, is that if there is a heart attack or you're looking at liver damage, one of the key enzymes that becomes a high concentration or higher in the concentration of the blood than normal is the aspartate and the alanine aminotransferase. And they only are found in the blood when cells are dying and breaking apart. These are easily detectable and are markers that are measured when you take routine blood samples. And so looking for these particular enzymes indicates somewhere there is cell damage that's taking place. And so you don't want to have these in your bloodstream. They are supposed to be in the cells as part of the metabolism, but that's another reason why you need to know what the aminotransferases are. Okay, once we have the amine that is released and placed into glutamate, what we really want to do is get the amine as a waste product. And so glutamate has a specific enzyme, glutamate dehydrogenase, that will react with it such that the actual amine itself in the form of the ammonium ion can be released. And so what we do is we have an oxidation reduction around the amine. And if I articulate that, what we're going to see is I'm going to take and make that a 2. All right. And we have an NAD plus as the acceptor. And so I'm going to go ahead and say there's a base, takes the proton, I'll form the double bond, and a hydride will leave and be accepted by the NAD+. We get another Schiff's base intermediate where we have a double bond nitrogen to a carbon. We know that the Schiff's base can be reacted with water, and we can show what will happen if water is deprotonated. It attacks. This can pick up a proton. Then we get an intermediate that has the carbon that was the double bonded nitrogen. We got the R and the R on either side. What we've just made was a hydroxide in our mechanism. Sorry. I'm going to put the hydroxide on the other side because that's where I wrote it. If I put it up there, it'll confuse everybody. So we have this nitrogen that now has 2 protons, no, 3 protons, as we saw, and the water attack left a hydroxide. Second step, the base takes the proton. I form the double bond of the ketone, and the ammonia is lost. And we get alpha-ketoglutarate back. This enzyme is the only, well, one of the only ways in which the ammonium is released, and that's important because ammonium in biological systems, in our biological systems, is poisonous. The enzyme can use NAD+ or NADP+. That's usually not the case in these types of oxidation reduction reactions. It is regulated by energy charge. So if you have lots of energy, ATP, GTP, it inhibits because I'm not going to want to degrade the amino acids for energy. And if I have indicators that there's low energy in the system, the diphosphates, it'll upregulate it through an allosteric effector. Most of the amino acid groups get deaminated by aminotransferase, but two have the ability to directly deaminate, and it's because of their structures with the hydroxides. And I'm not going to look or ask you about the mechanism. It's a little bit more complex with the cofactors that it uses, but you can see that if I lose water between this hydroxide and that proton, there's the water, I'll get a double bond. I'll eliminate to get the double bond, which is here. And then if you add water across the system, you can get the pyruvate with the amine falling off. But what you really need to have done is convert this into where this is a double bond and this picks up a proton before that happens. I can do that by deprotonating. And then you have a Schiff's base and a reaction exactly like we just looked at it. So serine that has that hydroxide to eliminate, and threonine, which has a hydroxide that can be eliminated, can facilitate this. So just a few other ways in which amines can be released immediately. What do you need to know about this slide? Not the mechanism, but these two particular amino acids are ones that do not have to use aminotransferase, although there are aminotransferases for these two amino acids themselves. All right. Once you have the NH4, we're going to get into what we've been trying to get to, is the urea cycle. And so we're going to produce urea as a waste product, and the alternative is the amino acids might be broken down in such a way to take that nitrogen and use it as a biosynthetic precursor to other compounds. But what we're going to use is we're going to outline the urea cycle for the waste product. And so this is the first metabolic pathway to actually be elucidated. All right. Overall, this is what happens. Okay. So we've got the urea cycle right down here. And so we're going to show that going on. Most of the amines are brought in from the peripheral tissue as glutamine. Okay. You can also degrade the internal amino acids, and then we can go to glutamate. What we're really going to get done here is incorporate this amine into the starting material, carbamylyl phosphate. And carbamylyl phosphate will enter in the urea cycle, will go all the way through, and we will get urea as the waste product. And so we're going to see aspartate as part of this process because it is going to be one of the reactants. So urea has two nitrogen. One comes from the general ammonium. The other nitrogen will come from an aspartate group. The overall cycle proceeds with four intermediates. Some of the reactions occur inside the mitochondria, and some of the reactions occur inside the cytosol to complete this cycle itself. And so the conversion of the amine into the starting material, carbamyl phosphate, is inside the mitochondria, and then we do one of the reactions in that particular compartment, and then the third, fourth, and fifth reaction where urea is released in the cytosol. The intermediates are ornithine, citrulline, arginiosuccinate, and arginine. Of that, you probably only know arginine at this moment in time as being familiar. All the other intermediates might be new. The aspartate, you know because it's an amino acid. And then the fumarate is what cycle? Citric acid cycle. Although it's being created in the cytosol, so it has to be transported back in. All right. In this particular case, the bicarbonate is a product of CO2 and water reacting together. It will be phosphorylated, and what phosphate do we attack looking at the reactant bicarbonate and the ATP? Alpha, beta, or gamma? Gamma. And so we've placed on this a phosphate to activate the carbonyl, allowing the ammonia, or you can also see it as ammonium. The pKa of ammonia is going to be protonated as it floats around in the cytosol. When it gets into the active site of the enzyme, it can get down to the NH3 form. The NH3 is deprotonated to act as a nucleophile, and we place the amine onto the carbonyl. The last step is to phosphorylate the acid component, and we've got the starting material. What is important about this cycle here is that to get the nitrogen in a form to try to convert it into a waste product, we actually use two equivalents of ATP to do this. That's quite a lot of energy to create this waste product. All right, the urea cycle itself. All right, carbamyl phosphate has our original nitrogen that came off of all of the amino acids everywhere else, and we're going to react it with ornithine. Now, classify what type of molecule ornithine fits into. Is there a class of biological molecules that ornithine is similar to? It really looks a lot like an amino acid. From our definition, it is an amino acid. It's got an acid, a nitrogen, a hydrogen, and an R group. This is just not one of the 20 that is associated to a transfer RNA. And so we talk about the 20 amino acids or the 20 common amino acids. There are lots of amino acids that are available in metabolism, but only 20 that are incorporated into proteins directly. So ornithine is an amino acid. All right. We've got our first reaction, and we had a volunteer left over from last week. Come on up. All right. And so I'll articulate something for you. Before we start, I'm going to go ahead and put a hydrogen, and a hydrogen. All right. And then in no particular place, we'll put a base. All right. What might you do with this?
Okay.
Now, fortunately and unfortunately, they will not be color-coded on the exam. One black and white is much cheaper.
I'm going to say, wait, hold on. Let me make sure about this. Okay. So, that grabs
Yep.
hydrogen, and then that would make a double bond on the nitrogen that can attack that.
Yep, the carbonyl.
And then you kick up.
And then you kick down, and
That goes, oh, wait, hold on.
yeah, if that breaks, go ahead and hit, yeah, there it goes. The electrons, when that breakage occurs, goes onto the oxygen, and you have the pi leaving group. And so citrulline is your product. That's perfect. Now you can select the next person. Okay, who has not done this before? There's still a group of people that have not come up. We got the back row, all in the back out there. I think everybody over here, so you've got choices. All right, if you take a look at this, ornithine transcarbamylase, what we're doing is we're transferring the carbonyl with the amine, and the phosphate is the leaving group. And so we've created just a little bit larger molecule, but do you see urea as a possible product at this moment in time? Okay, come on down. All right, at this moment in time, we have 3 reactants: citrulline, aspartate, and ATP. Now, very rarely do 3 reactions occur at one time, or 3 reactants, and so we're going to choose 2 of them. We're going to do citrulline plus the ATP as the first reaction to create the intermediate that is drawn. And so, what in the world happened here? Okay, so in that, if you're taking a look at this, well, let's choose this. You see this one right here, right? That nitrogen. And over here, that nitrogen has how many protons?
2.
And how many does it have here? 2. Let me articulate something for you. Oops. That was not exactly what it, but there are a pair of, oh yeah, go ahead. There are a pair of, lone pair of electrons on that nitrogen, okay? And you see in the product the nitrogen has a double bond? Okay, so our first step would take that lone pair and make a double bond. That's weird, but yes, it does. There, okay. And so, as you make that bond, what happens is the carbon, where the carbonyl is, has 5 bonds, right? So, something has to kick up, okay? And that now is sitting on the oxygen, but that now makes the oxygen an electrophile. And so, why don't you hit the back arrow, or the back right next to that little circle there. Okay, all right. And so, that pair of electrons can attack somewhere. Where might it attack?
The ATP?
Mm-hmm. And so, on this ATP, where is it going to attack?
The alpha?
Yes, it attacks the alpha.
Do I have to exit?
Yeah, go ahead and attack. Try to get it close to that alpha. Okay. There. Boom. And then electrons do what?
Kick up, kick down?
Kick up, kick down, as best as you can on that little, on this little thing there. Kick up, ish. Okay. Kick down, and then it's hard to articulate because it's really tiny. There. Okay. And for that electron movement, all of them have to have an arrow, arrow, arrow. And then I can't even get an arrow in that little tiny thing. But you have done the first reaction. Go select some other volunteer. I won't make you do 2. Dun da da da. Oh, my goodness. What's going to happen here? Okay, I'm going to simplify something for you so we can simplify the mechanism. All right, it gets into the active site, and it's going to deprotonate 1 because it's in the active site. So it'll only have 2. It will not be charged anymore. And I will just write a base, any particular place on that. Okay.
Oh, I did something.
You did something. What did you do? Okay. There you go.
Okay.
Okay.
Alright, I won't touch it. Alright, so it only has 2 right now?
Mm-hmm. It only has 2 right now. Okay. Good.
Let's take that. Let's go.
And that's going to attack. Yes. Okay.
This is gonna do that. Let's say.
Okay. Anytime you have a double bond, you're going to have to, when you attack, has to kick up, kick down, and yep, the AMP falls off as the leaving group. And you got that back. Now, what is this? Oh, wait. Go ahead and choose somebody. I'm going to use the pencil for a second. What does this represent right here? Anybody? What does... Yeah. So we've got resonance of that double bond. All right. So when he drew his mechanism, what he left was the double bond in this position, but it's roaming around. All right. So there is a double bond in all 3 of those positions. But at some point in time for the next reaction, we're going to have to lock the double bond into one of the 3 conformations to proceed with the reaction. Okay? And so we have now arginiosuccinate, made up of 2 words. What 2 words? Arginine and succinate. Okay? And so we're one away from getting arginine and succinate together, and then one step, we're going to get fumarate. All right, you ready? Okay, we always have a base to use, and we always have an acid available to us. All right. Now, as you look at this one,
So this would gain a proton.
which one gives a proton? Yeah, the nitrogen that's in that conjugated system has to pick up a proton, yes.
So this would attack here.
And so there's something else. There's something else that's going on too.
Would this electrons go to make this double bond?
We have to have some electrons to make the double bond. Yes, that's correct. Okay, so let's take some inventory real quick. All right, right here, no protons, right?
Yeah.
Right here, 1 proton. Right here, how many protons?
We have
Understood.
2.
2. This is the key to this one. And so now if you see this, carbon, carbon, carbon 1 hydrogen, carbon 1 hydrogen, carbon 1 hydrogen, carbon 2 hydrogens, and then that one. So do you see where we're going to start? Oh, let me get the color.
Does it matter which proton has?
No, because they're equivalent. Okay. Okay. Now, okay. Which hydrogen did you attack?
Would attack this one.
Okay. Then wait. Go hit the back arrow. So go ahead and hit that. Don't attack the electrons,
Okay.
because that's what it looked like. All right. I thought, there you go. Now the pair of electrons on the other side of that hydrogen can go where?
To the double bond.
Yeah, show the double bond.
So from here.
Yes. Now, a pair of electrons around that carbon now has to leave. Which one does the leaving?
So would this one leave?
Yes, and when it leaves, it can then go pick up a,
A proton.
that's correct.
Okay, so do I just attack from here or just one?
Yeah, you can attack from right there. Okay, so the pair of electrons just picks up the proton. And so what happens is, this pair of electrons picks up that proton, and that becomes nitrogen with 2 hydrogens. Nitrogen with 2 hydrogens. And then the double bond is this double bond, and you've done the reaction. Perfect. Go select the next person. All right. I have the carbon chain of fumarate that has left, and we have this now arginine. And so arginine. The next reaction is going to be arginase, the enzyme, so I'm going to break it down with water. And so who's going to come and break it down with water? All right. As a class, where are we going to put the double bond? All right. First position? Got it. Second position? Or third position? Where will the double bond be fixed so that we can get to our product? You say third. Okay. Anybody else want to? You would agree? Okay. Let's lock it in the third position just for this reaction. Okay. And then we have water that is available, and I didn't articulate that. Okay, and we have a base. Okay, and then we have a proton.
So the base is going to take a hydrogen from there, and so that'd be like O minus, and like O minus would come here and attack.
Yep.
And then, see, the double bond is there. So wouldn't that just lead you to your intermediate where you have the OH? We have
Yep. That's it. You've got the OH attached. You've got both of the nitrogens attached. And, oh, there's one thing you have to do.
oh, you have to grab a hydrogen. So it's, ooh.
Oh, you hit.
I don't know how I did that.
You hit the undo.
Oh, just kidding. Okay. So, please grab that.
Uh-huh.
There. And then it's from the double bond.
From the double bond, yes. And then that movement allowed you to make this portion there. All right, go select someone else. Anybody that hasn't done it? Is there one more? Your choice is locked. All right, the last movement, we're going to produce the urea. So what can be reacted to form that ketone in the urea?
The acid.
And so, all right, let's do some inventory then. Okay, this acid is that one. There's a carbon, there's the carbon, there's the nitrogen, there's the nitrogen, there's the hydrogen. So we follow up the chain, and we're supposed to end up with a nitrogen that just has 3 protons. We have a nitrogen that has 2 of them. Okay? On this side, we got a carbon. It's got 2 nitrogens attached, carbon, 2 nitrogens attached, and then an oxygen, but in the wrong form here. And so what we have to do around this oxygen is get rid of what? The hydrogen. Okay? And so you found where you're going to do your first. Yes, select a color. Okay, and so did that form the double bond? Yes. Now, you got too many bonds to that carbon, so the bond between the nitrogen and that carbon has to leave and pick up the proton. Beautiful, perfect. And you've created urea. And we've created ornithine back. And so it is a cyclic intermediate, or cyclic pathway. And so we have a very concentrated amount of amine in a waste product that's very soluble. And we all know what the color of urea is, right? What's the color of urea? It is not. You would think it is. Urea is clear. And so if ever urea is quite often used in laboratories to denature proteins. 2 interesting things about urea. You think it's yellow. But when you dissolve it, it actually becomes cold. It's an endothermic reaction when it dissolves in water. And you have to make a 8 molar solution of urea to do denaturing. And so when I do a demonstration of endothermic processes by dissolving urea in a tube, I pass it around and it feels cold. Quite often, cold packs have inside of them urea salt. When you open it up, open the water in there, it'll form the cold. Overall, the equivalent of one carbon dioxide, it was in the form of bicarbonate, 1 amine, 3 ATPs and aspartate, 2 waters yields a urea, and a lot of energy that is used. Also fumarate. And so you have to have aspartate coming in into the reaction as part of this as a reactant. Fumarate is part of the citric acid cycle. Now, the urea cycle and the citric acid cycle are linked between a few of these reactants. It's called the aspartate argininosuccinate shunt, or also known as the Krebs bicycle. And so what we just looked at was the conversion of carbamylophosphate with ornithine into citrulline, citrulline, argininosuccinate, and we just did this urea cycle. We said our aspartate came in and reacted, and we said fumarate came out. Okay, fumarate is converted into malate, which is a citric acid cycle reaction happening in the cytosol. Then it is transported inside the mitochondria. We get conversion of the malate into oxaloacetate, which is part of the urea cycle. I'm sorry, part of the citric acid cycle. And the oxaloacetate can be taken and a transamination can occur and I get aspartate, and then that aspartate comes here out to react. And so this is how the citric acid cycle and the urea cycle work together to allow degradation. Now, here is a waste product in terms of the amine coming off amino acid degradation, but here amino acid degradation also funds that second amine because the oxaloacetate doesn't have an amine, but amino acid degradation will give it to glutamate, who's the carrier, and put it into aspartate for just a period of time such that it can form the urea. The carbon chain comes back. And so I am getting 2 equivalents of waste amines out of the system through this cycle and through the working of both of these. All right, and so oxaloacetate becomes one of those key structures that have a lot of different ways in which it can go. It actually is part of or funds the urea cycle. It can go through and be used for gluconeogenesis. It can also be used through the citric acid cycle. We haven't even begun the uses of synthetic processes that that particular molecule can be used for. But in degradative, there are a lot of different ways that oxaloacetate can be used. Now, sometimes there is a defect in the urea cycle, and that's not great. Okay? Blockage of it can be fatal because I told you ammonium is poisonous, and it's poisonous at the 60 micromolar level. Okay? If you go into the laboratory and you try to make a micromolar concentration, it is very, very difficult to do. You're going to calculate something in just a few milligrams in even 100 mils. And that's hard to measure out on a scale, on the balance, and get a good measurement. So a lot of times when you're in biology and you have to make a very, very low concentration, you start off with a very concentrated, maybe 10 times the concentration. You make a really small amount of that, and then you dilute it. But 60 micromolars is a very low concentration before it becomes toxic. A lot of times there is a deficiency in the argininosuccinase, the enzyme that creates the arginine and the fumarate. And so what can actually happen is that CO2 and the amine can be incorporated together, can go through this cycle up to this point, okay, but it can't go past this. Consequences. I don't have arginine being produced, I don't have urea being produced, and I don't have ornithine being recreated because it's a cyclic intermediate. Now, at some point in time, if this is what's happening and this doesn't work, you'll use up all the ornithine and you'll have lots of this, and that's not necessarily great. All right, treatment is to do this. We treat by adding lots of this intermediate, and so we can create ornithine as a precursor for the cycle. The waste product becomes this molecule. The kidney doesn't quite like it, but it will prevent toxic buildup of the amine. Why in the world? Okay, so we add arginine. Why don't we just add this molecule? Wouldn't that be better? You wouldn't produce urea, but really the urea that you're getting off here comes from what you give, comes from the drug you give, because your waste product who cares? Okay, because your waste product is the arginiosuccinate, right? So that urea is just part of what you treat with. Why in the world do we give arginine rather than ornithine? Both would work as drugs, absolutely. Yes, our arginine, we don't even have to make it, we just have to isolate it. We don't have to synthetically make arginine. Now ornithine, on the other hand, there's no great source biologically because it's always being used in the cycle, and so ornithine would just be more expensive. Arginine can just be isolated from natural products, and so it's cheaper. And so sometimes it comes down to the dollar. All right, other issues. You can have deficiencies in either incorporating the amine into the carbamylyl phosphate, or even if we do get in carbamylyl phosphate, we can't react it with ornithine. All right? What we do in this case is you limit the intake of protein. If you limit the intake of protein, then even turnover may use those amino acids to incorporate the amine into synthetic precursors rather than breaking them down. What we will do is add benzoate and phenylacetate as drugs. What this will do is react with the molecules in which, the amino acids in which the amine actually builds up. Quite often, the muscle will send out glycine if there is a large influx of breaking down of protein. And so the glycine can react with the reacted benzoate. And the glutamine is the natural molecule that builds up lots of amine, and so the phenylacetate will react with it, and so these two molecules are excreted. So what we do is we use drugs as the incorporation. Rather than incorporate it into urea, we incorporate it into these drugs. You would have to take these all the time. If you actually look at any packaged food, quite often you see benzoate as part of the process. Benzoate is a preservative, and sodium benzoate is most often used. So it is something that we do ingest. It hasn't necessarily hurt us too much, but this is how we would bypass that particular defect, or those two particular defects. All right. So we've taken care of the amine itself. We're now going to start taking care of all of the carbons that come off as alpha keto acids. All right, there are 20 amino acids. So that means there should be 20 entrances into these pathways that break things down. Things that I want you to know: what kind of pathways are used to break things down? Obviously, the citric acid cycle. That's a huge one. Okay, and we see the ketone bodies as a pathway by which things are broken down. And we have the entrance of pyruvate, which is the end product of glycolysis, but we use the first step of gluconeogenesis to that can enter in the pyruvate and or go through the citric acid cycle. Now, if you count up all of the amino acids and where they enter in, there should be 20, right? If you start counting them up, what happens? Okay, there are, what, 1, 2, 5 here, plus 3, 8, plus 3 is 11, 12, plus 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24, 25, 26. There are 26 entrances. If you take a look at some of them, they have multiple entrance places because they're large carbon chains. And so in the degradative process, parts of them go into different places where degradation happens. All right? And so we will start looking at degradation of amino acids. Now, the book itself describes certain number of groups and where they go for. I'm going to describe 5 groups and then 1 oddball, leucine. And so we're going to account for 19 of the 20 amino acid degradations into kind of places where they go. And so group 1, they indicated smaller amino acids go into pyruvate, and then parts of the large amino acids go into the ketone bodies. Then we have a group 2 in which a group of those amino acids tend to go to alpha-ketoglutarate. Then we have group 3, the aromatic amino acids. Most of their carbons go into acetyl-CoA. Then we've got a group that will send their their carbon chains into succinyl-CoA. The asparagine aspartate go to oxaloacetate or fumarate, and then leucine has its own degradative pathway that's not related to any other amino acid, and we are in the ketone bodies and in their degradation. And so you can learn the amino acids by where the majority of their carbons enter into the degradative pathway. So this one's an important slide. Now, group 1, lots of reactions. And I just love this kind of slide. Many reactions later, you can see where they will all kind of end up. Very few of these kinds of reactions we're actually going to be able to really look at in detail. We will take a look at a few. But in these cases, what I really kind of want you to know for the overall each amino acid, where the majority of their carbons will end up. And so this overviews those that go to the pyruvate. So if I take a look at a couple of the reactions themselves, all right, we have 1 amino acid that is sitting up there. And can you name what that amino acid is? The amino acid that we start with. I didn't actually put its name. Serine. Okay? And so we're going to look at the serine reaction that was on this slide. Serine was somewhere on the slide. Where is it? Serine. And so we're looking at the degradative process of this one. Okay? So first step would be a oxidative step in which NADH is made. To do this, if I look at inventory, the reaction centers around this area where I have a hydrogen and a hydroxide. I now have a ketone. So I will use a base, take that proton, create a double bond, and a hydride leaves to create the product where we have a ketone. That one step is as much of a question for mechanism as I would ask you to do. Not an entire slide like this. All right. The next reaction is kind of a throwback to the enzyme that uses PLP or the aminotransferase. And what can happen in this is the amine reacts with the PLP and the steps that we saw that began the aminotransferase. And what actually happens is we have a carbon of the functional group double bonded to the nitrogen. All right. The next set of reactions that occur would be something that I would expect you to know, the conversion of this thing to here. Now it looks scary because it's got all of this organic stuff around, but if you take a look at inventory, the carbon here and the carbon here doesn't change, okay, that methyl group. But around this ketone, here's a ketone, and here is, whoa, not a ketone anymore, an oxygen with a negative charge. It is now attached to a CoA. All right? And so I can see the rest of it is not changed. So if I gave you the starting material, this, and I gave you this is the reactant, I would expect you to be able to take, and let's take off the proton, let's allow the sulfur to attack, and then we'll kick that up as a negative group. Okay? What that is a transition state with a high energy component because it's in the negative charge on the oxygen. All right? And then I could and would be very comfortable to allow, if I basically wrote this to this, with this as the outcome, I would expect you to be able to do that. If you take a look at this, we know that this acetyl-CoA comes off. All right? I see the formation of acetyl-CoA with the oxygen here, but I got the wrong state. Also, if you take a look down the line, this acid is that acid. There's a carbon, there's a carbon. There's a difference in the double bond there. The double bond has moved, and it moves all the way through. So the mechanism is going to take this pair of electrons to form the double bond of the acetyl-CoA. This is the pair that leaves to form the new double bond, which then moves this. Whoa, that's not an arrow. I don't know what that was. That is an arrow, which then allows this pair of electrons to fall here and that nitrogen to take the lone pair. The whole reason we looked at that whole functional group in transamination and in this particular mechanism is for that movement to occur. It allows this bond to be a leaving group because this resonance form is available to it. And so that carbon to that carbon, breaking a carbon bond is difficult because you normally are left with a carbanion. That's not great, but if you have this cofactor, it allows that carbon-carbon bond to be broken. And then I have acetyl-CoA falling off, and I have the rest of the molecule that is attached. All right. I can go back, and from here to here, you see the ring doesn't have is not aromatic. The ring is aromatic, so I have to do some changes. Here I have a double bond. Here I have a single bond. Here I have one proton. There I have two. So the mechanism must include the lone pair falling back, this pair falling back, and this bond picking up the proton. All right. All right. As we finish this up, what can occur in this particular next step is to deprotonate. The electrons can fall here to create the double bond you see in the product, and then this one picks up a proton so that you have two protons there. Ooh. All right. The last step, when we brought in this particular molecule it was bonded to a nitrogen of the enzyme. The nitrogen of the enzyme is going to attack, this will kick up, and it'll eliminate, and this is the falling off leaving group. What amino acid is this one? That's serine. What's this is down here at the bottom? Glycine. So, serine can be broken down into acetyl-CoA and glycine. The mechanism for this, if you're interested, is the same mechanism as the aminotransferase. The mechanism for this reaction is the same mechanism as aminotransferase. It is multi-stepped. And so I wouldn't expect you to do 4 or 5 steps in any particular question on an exam. I would look at one step to the next, and not a multiple step, 3 and 4 step process. All right, so serine can be acetyl-CoA and glycine. Group 2, again, overall picture, histidine, arginine, proline, all of them go through a pathway of degradation until we get to glutamate. And glutamine can be deaminated to become glutamate. And glutamate has a special reaction with a dehydrogenase to allow the amine to fall off as NH4. And then I have alpha-ketoglutarate, which is obviously the citric acid cycle. So all of these will allow their amino acids to become alpha-ketoglutarate, even though they're quite different. Now, we did see urea, arginine giving off urea going to ornithine. Ornithine normally goes in the urea cycle, but ornithine does have a methodology to degrade all the way down. We don't normally allow this to occur unless we have an overabundance. Most of the time, the ornithine that you have stays in the urea cycle, but we can degrade it if we need to. The group 2, this is the biggie, and we've looked at the mechanism prior. Oxidation to create the double bond, adding water across this to get the alpha-ketoglutarate. We showed the mechanism at the beginning of this lecture. If we look at group 2, we're going to look at arginine and proline being degraded. And so we're actually going to get to glutamate through a common intermediate, which is glutamate gamma semialdehyde. And so proline is kind of oddball. It's got the ring, and you would think that would make this really a more difficult amino acid to degrade than it really does. But the first thing is an oxidation. And we're going to take a look at the product here and the reactant, and we can see around this bond that it becomes a double bond. I should only have 1 hydrogen and 1 hydrogen. I have 2 and I have 2. I know where they're going to come from. The question would be, which amino acid, which proton do I deprotonate? The protons on the carbon or the protons on the nitrogen? Which would be the one that gets deprotonated? The other one's going to be the hydride leaving. Carbon's deprotonated. All right. If I deprotonate the carbon, then the electron pair sits on this carbon for just a moment. Okay? That's a carbanion. If I deprotonate this, for a second, the electrons can kick onto that nitrogen. The nitrogen would have a lone pair. The nitrogen would be neutral. Which is better in energy? Nitrogen. So we're going to deprotonate the nitrogen. The electrons will form the double bond. The leaving group will be the hydride. I can only do that because I have an electron acceptor. Okay, double bond. Took off a hydrogen, and in the intermediate, I'm going to add water. To add water, to create that hydroxide, got to make a hydroxide, deprotonate the water, becomes a hydroxide. The negative hydroxide becomes a nucleophile, attacks at that carbon because the oxygen is attached to that carbon in the product, and then I go ahead and pick up the proton. All right, the last reaction. Okay, I'm going to make an aldehyde. Okay, well, I've got an oxygen, I got a hydroxide, but I want an aldehyde. So let's take that proton away and do something with the electrons. If I take that proton, the electrons can form the double bond to create a ketone. And what can occur is this nitrogen can be the leaving group to pick up a proton. If you do inventory, this nitrogen here, okay, is this nitrogen there. So that's the nitrogen that is the leaving group, and this hydroxide was will become the ketone. So proline is now at glutamate gamma semialdehyde. Sorry, proline went to gamma semialdehyde. Here we have arginine. Arginine can react, and we did this. We already saw the oxygen reaction with arginine in this urea cycle. We had this intermediate in the urea cycle, and we made ornithine in the urea cycle. What the leaving group is, shoot, I didn't write it, urea. Okay. The new reaction that is the next reaction that we see is the conversion of ornithine to the glutamate semialdehyde. Oh, interesting. Okay, let's do this real quick. 1, 2, 3, 4, 5, and the nitrogen. 1, 2, 3, 4, and 5. That's weird. I get that nitrogen that is attached, that carbon that's attached to a nitrogen becoming an aldehyde. All right, unfortunately, I left off some intermediates to get to that point. What we will do is, one step to get here, is we've got to make this intermediate. Okay. Okay, and so if we get this intermediate, is that, no, that can't happen. We would have to have an oxidation to get to that. I don't know why I didn't show that. Take that away, take that away, take that away. We would have to get to too many takeaways. We would have to get it to this intermediate where there's a Schiff's base. And if we have a Schiff's base, then we can add water and get to the semialdehyde. So I need to correct this slide so we can show that, which we are not going to show for this lecture. All right. Once we have the glutamate semialdehyde, we actually need to make glutamate. And that is not too hard by adding water. Gets a really kind of weird intermediate. A dialcohol right there. So an alcohol on the top and alcohol on the bottom. It's neutral. It's an intermediate state, but that's not something we normally see. But by doing the oxidation, we get a very stable intermediate because we create resonance structures. Deprotonate that one. Doesn't matter which because they're equivalent. And we create the glutamate. And so both arginine and proline create glutamate. And then glutamate can be converted into alpha-ketoglutarate using the dehydrogenase, glutamate dehydrogenase. Histidine, on the other hand, looks really, really strange. Okay. And in this, how would we get the double bond in this position? Anybody tell me what to do? Deprotonate. The one, which one? This one? Nope. This one? Yep. If you did inventory, you see that there are 2 protons there and 1 proton there. Now, one good thing is you do have the ability to have a leaving group. And so a base in the enzyme can allow that to fall off. And I get a really strange molecule. This looks easy. It is not. It actually reacts with NADH, NAD plus, but in no way near what you might think. The reaction occurs with the place where a hydride is usually accepted, which is right here. And what happens is you take the lone pair and this pair of electrons attack and attach where a hydride should be accepted, and then you do through all of this to get your ketone. And so what I show here is NAD plus actually does other reactions than just accepting a hydride. I don't want you to know this particular mechanism. I just wanted you to be aware that there can be other reactions with NAD+. Okay. In this particular, this would be a fair mechanism to ask of you. Okay. The water adding to this particular molecule to create the product. What might you do with this one? Oh, good. We'll deprotonate the water. Okay. Usually when you add water, when you have water, that's usually the first step. Okay, now I have a hydroxide. It can be a nucleophile. Okay. Now, this is just like the test where it's not labeled, but if I label it for you, one of these oxygens here is the oxygen from that water. All right, I attack the ketone. Then what do you do? Kick up, kick down, and then what happens? The bond between the carbonyl and the nitrogen will pick up a proton. Essentially, what I've done is added water across a peptide bond and broken it. And so that kind of slide, that kind of mechanism from the beginning to end would be the type of mechanism that I could ask you to reproduce on an exam. Okay. Oh, interesting here. Do you know what THF is? Have you heard of THF before? It is an organic solvent, yes. The first time you've ever seen THF probably is tetrahydrofuran, and it's an organic solvent, which is totally confusing because in biochemistry, this is called tetrahydrofolate. We'll take a look at that next semester in much, much more detail. It is a molecule that will carry 1 carbon intermediates, and so it's a really weird reaction. It's going to grab that carbon and take it away. and it takes away whatever's attached to it, which is that nitrogen, and then we've got to glutamate. And so all of those, histidine, proline, and arginine, are all converted into glutamate, which then is converted into alpha-ketoglutarate. So we have finished group 2. Whew. All right, group 3. Interestingly enough, what we're going to do in this group 3 is know that all the aromatic amino acids will end up as intermediates of the ketone bodies here and fumarate, which is the citric acid cycle. And so that's the ultimate thing that you need to know, that the phenylalanine and the tyrosine both create intermediates in the citric acid cycle and ketone bodies. Interestingly enough, tyrosine can be broken down or converted into dopamine, epinephrine, and the melanins. What do you know about dopamine by things that you might have studied before? Have you heard that name? It's a neurotransmitter. Okay. Epinephrine, you've heard of that, right? So an amino acid gets converted into that hormone, and then the melanins are the synthetic process to pigmentation. And so tyrosine is a really kind of interesting molecule in which it can be degraded and converted into lots of different things. Phenylalanine, its conversion is all phenylalanine gets converted to tyrosine in its breakdown product. So we become convergent at that point in time. In this case, there is a additional electron acceptor, and this is important. We know that we've been introduced to 3 electron acceptors in terms of mechanistic reactions. The NAD+, the FAD, and NADP+ can accept electrons, and in special cases, all of them can donate. In this case, the tetrahydrobiopterin is another very specialized acceptor, and so we have 2 different ways in which the electrons can be accepted and or donated to this acceptor. And so this is an electron acceptor used in the degradation of these 2 amino acids. And so knowing that we have 1 more electron acceptor is important about this slide. Not the structure, not where it actually accepts the hydride, but that they can accept hydrides and they can donate hydrides. And so really interesting reaction with the acceptor. Most of the time when you see hydroxylases that react with oxygen, there's a iron center that coordinates to all of this. It's really complex. I don't want to get into the mechanism, and you don't either. But what we add is the hydroxide. Tyrosine will be converted into the organic alpha-keto acid by the use of the transamination. And so you guys can go back and actually look at the mechanism for transamination, but this is now the carbon chain I must get rid of. Again, some reactions that I'm not going to necessarily require you to know. The organic molecule reacts directly with oxygen. It creates a per acid. That's an unusual organic structure, which is very reactive, which then creates an epoxide, which is really reactive. And not only do you have the epoxide formation, but then you have a carbon shift to get to an intermediate. Reactions that are not necessarily ones we will go over. This reaction is similar to the reaction called ozonolysis, where I'm going to break a double bond and create 2 ketones. I will create right here and break this double bond, creating a ketone and a ketone. And so that ketone are created by the breakage of that ring. In terms of the reactivity, it is highly energetic because not only are you breaking it down, but you're also breaking aromaticity, and so it requires a lot to get that done. No mechanistic, I'm not going to ask you about a mechanism for that. We have a isomerization, just creating a conversion around the double bond. This would be a slide that could be a type of mechanistic reaction that I may ask you on the exam. All right, there's one thing missing that I would give you on the exam, is the addition of water to get this done. All right. If you have water adding, the mechanism begins with deprotonating the water. Hydroxide is a nucleophile. Where will it attack to end up in the product? The carbon with a red O. If you do kick up and kick down, the leaving group can be this black bond which picks up a proton. This carbon has 2 protons. When it picks this up, it has 3. Becomes this methyl group. Okay? And when you have finished this part of the reaction over here, you actually end up with a double bonded O to a hydroxide, an acid. It's shown in the deprotonated form. Okay? The last part of the question, what pathway are we here with these carbons? The which one? Citric acid cycle? The acetoacetate? That's not the citric acid cycle. What pathway is this molecule a part of? These are all open test questions. It's a ketone body. And then fumarate, what pathway are we in? That's a citric acid cycle. So, big picture, phenylalanine, tyrosine, citric acid cycle in the form of fumarate, and the ketone bodies, acetoacetate. All right. In this, there's not a lot of questions I could ask you on the exam in terms of mechanism because they are too complex. Okay. There is a disease that occurs in terms of the phenylalanine's degradation. And so if you have an impairment of the hydroxylase that converts phenylalanine to tyrosine, then you've got problems. What can occur is the phenylalanine starts to react with pyruvate to form alanine, so it gets rid of the amine, and we get 2 metabolites, 2 end products, the phenylacetate and the another alternative that are products that are formed in the bloodstream and tend to compete with neurotransmitters, and so you have neurological deficiencies if you are not able to degrade the phenylalanine. And so, in this case, there is really not very many treatments that you have available to you. Really, the only thing that you can you have to watch out for is intake of this particular protein. So people who have this particular absence have mental retardation and shorter life expectancies. And what really happens is myelination of the nerves is defective. If you can catch it early, you can prevent it, and the only treatment that we have is to limit protein intake, and particularly the phenylalanine and any of its derivatives. So there is one of the artificial sweeteners that has phenylalanine as a base for its synthesis, and you can see sometimes on packaging they'll warn people not to take that if you have this deficiency, because that's a very concentrated amount of the wrong amino acid for them. Aspartame is the artificial sweetener, and so when it breaks down and in the body it creates aspartate, phenylalanine, and methanol. All of those are normal byproducts that we can get rid of other than methanol. Usually don't drink methanol too often, but it can be degraded, and you can see that there is packaging requirements if there are components that will break down into phenylalanine, so people know. Okay, succinyl-CoA entry. So the type of amino acids in group 4 all enter in. The majority of their carbons go through succinyl-CoA. And here we find methionine, valine, and isoleucine will create propionyl-CoA, and propionyl-CoA goes into the methylmalonyl-CoA. We've actually seen this pathway. Where do we see this conversion? And it requires a vitamin. Requires a very big vitamin. No, it's not the ketone bodies. Yes, beta oxidation of odd numbered carbons. Because when you do all of that, if you had an odd, you will always end up with a propionyl-CoA at the end. And we said we had to have what vitamin? Not biotin. Vitamin B12, which is cobalamin and has a really weird mechanism, okay, where it will convert it into, succinyl-CoA through that vitamin. So I told you that we very rarely eat odd-numbered fats, but this is where that vitamin is more important, is the degradation of these particular molecules. All right. To get to that place, though, to get to this place where they all go through that, if you take a look at valine, it only takes 7 steps to get to here, and then 2 steps there, so 9 steps to get there. Threonine is a whole lot quicker. It takes 4 steps to get there. Isoleucine takes 6, 8 steps to get to that one. And then methionine takes 3, 4, 5, 6, 7, 8 steps to get to that. And so even though they all come to this intermediate, it takes a lot to get them. Big picture. Methionine, isoleucine, threonine, valine, go to succinyl-CoA. That's the big take-home picture for this particular group. What pathway are we in with the succinyl-CoA? Citric acid cycle. All right. Group 5. Aspartate and asparagine, both will be converted into oxaloacetate and fumarate. Okay, to convert asparagine into aspartate, we're going to add water. It comes off as ammonia, but then gets protonated immediately to the ammonium because of pKa. All right, the aspartate to oxaloacetate is through transamination. That's a big, long mechanism. You can look it up if you'd like to see it. Okay, but an alternative is to react it with base to get to fumarate. To do that, NH4. All right. What pathway? Citric acid cycle. What pathway? Citric acid cycle is 1/3 right. Or the urea cycle. Those two is 2/3 right. And the third one is gluconeogenesis. Now you've covered all of them. Oxaloacetate is a huge intermediate that goes to lots of different ways. Now, why would it go, it's got 3 different pathways. Why would it go down one of those pathways? At that moment in time, what does your body need? And it will shift to either one of those pathways depending upon what is needed. Most of the time, oxaloacetate will be part of the citric acid cycle. That's much more often used than any of them. Okay. Wait a minute. Am I seeing double? Okay. That slide adds not a whole lot to the whole process there. Okay. The reaction of aspartate through arginiosuccinate to create fumarate is part of the urea cycle, and so it doesn't necessarily have to go through the transamination. It also has a reaction. There's another pathway that gets it not to that one, that gets it here is also the urea cycle. I think that's why I added that slide in. Leucine doesn't fit anybody's group. Leucine is kind of its own group. And so first step is to take the amine off through transamination. Again, that's a long mechanism. And now we have the alpha-keto acid. This is an interesting reaction. And you should be able to see that it is related to 2 other reactions that you have seen in the past. If this carbon was just a methyl, then this reaction gave you acetyl-CoA. Okay, if it was, there's a reaction in the citric acid cycle that makes succinyl-CoA. It uses a large complex called a dehydrogenase complex with lots of cofactors included in lipamide and TPP to do that full reaction. And so we see an oxidative decarboxylation. This is decarboxylated, and we add CoA. It's related to those other 2 reactions. So this is a multiple step reaction, not something I would ask you to reproduce. But you should be able to see that it is similar to other reactions that you've seen in the past. Now this one, I would absolutely expect you to be able to do in the exam. We've got FAD going to FADH2. That means we know we're going to have a hydride. And if you take a look at inventory, we change around this particular bond, and it becomes that. When you see something different, do this always. Write in the hydrogens that are not written but are understood, because that might give you a clue as to what to do. If you look at the 2, we got extra pairs of hydrogens. Okay. So Too many hydrogens, too many hydrogens. So which one of these two would I deprotonate? Would I deprotonate this particular proton, or would I deprotonate one of these two particular protons? The alpha carbon. Because it is alpha to a carbonyl, they are acidic, and it's easier for a base to take them. The electrons will create the double bond, and the FAD will pick up the hydride. Okay, that is something that is very fair game. Now that reaction does something very interesting. Okay. All right. The CO2 here, I'm not going to show the mechanism, but the CO2 reacts with water to be bicarbonate, and the bicarbonate reacts with ATP to create that intermediate at the top. Okay? So this is the reactant here with this. What in the world do we have to do to this thing to react with this to get this product? When you look at the back and forth, what are you missing? A hydrogen on one of the methyls. So this one started with 3. In the product, it has 2. Now, is any of those protons acidic? Or is that methyl group acidic? Why were alpha to a carbonyl acidic? Because of resonance, yes. Is this alpha to a carbonyl? No. Okay. Watch this. If by chance I take one of those, this is not part of the mechanism, but I'm going to show you something. I could kick it in here, I could kick that there, and I could kick that there, and it is able to form a resonance structure with that oxygen. It is as acidic as an alpha carbon because of that double bond that is there. This is why all of those protons are acidic. Are these protons acidic? Just as acidic as these. Why in the world do I react only here? Yes, and that's kind of the answer for all biological systems. It's the enzyme. The base will only be around these protons. If by chance you allow this to react with this in an organic flask, you're going to have reactions here and there, most likely in about a 50-50 chance that either one will react. All right, the mechanism for the reaction is this. The electrons will attack the carbonyl. I'll kick up, I'll kick down, and the leaving group is the phosphate, and I will carboxylate or add an acid group. Okay. Ooh. What are we going to do here? We're going to add water to it, right? And you can see where the hydroxide has to attack. In the product here, we've got CoA-S, CoA-S, carbonyl, carbonyl, a carbon that has 1 hydrogen, a carbon that has 2, and then a carbon that has no hydrogens, and a methyl in this group. There's the methyl in this group, and there's the hydroxide. So the hydroxide has to attack at that. Okay, and pick up the proton. Okay. What pathway are we right here? We're actually in a pathway that we've covered. No, not the pentose phosphate pathway. Okay, got one down. Is it glycolysis? Gluconeogenesis. Citric acid cycle. Urea cycle. What other cycle? What other pathway did we look at? Beta, is that beta oxidation? Nope. This is ketone body formation. And this last step we actually looked at in ketone body formation was a formation of acetyl-CoA and acetoacetate, both of which are part of the ketone bodies. And then they can be degraded from there.